All right, hello everybody. Welcome to the HIRM seminar for the 15th of December, 2020. This is our last, I think it's our last seminar of 2020. And it's a great pleasure for us to have Dr. Ian MacRae with us today as our seminar speaker. Dr. MacRae is a professor in the Integrative Computational and Structural Biology Department at the Scripps Research Institute in La Jolla, California. He obtained his bachelor's at the University of California, Davis in biochemistry, and then his PhD in structural biology also at UC Davis, before joining Jennifer Doudna's lab as a postdoc at the University of California in Berkeley. And he has then started his own lab at the Scripps Institute as an assistant professor and has worked all his way up to being a professor there now. And he's one of the world's experts in the structural biology of small RNAs. And apropos that, his talk today is going to be called Small RNAs in Animal Biology, a Structural Perspective. So it's wonderful to have you here with us, Ian. The floor is yours.
Oh, thank you. Thank you, Frank. And thanks for everyone for coming. There's 70 people. This is really, a lot of people I recognize. This is really nice. Okay, so as Frank said, my lab studies small RNAs, and I'm a structural biologist. And so I'm going to try to describe how I think about small RNAs and how my lab thinks about small RNAs from this perspective of what the molecules look like, right? Shoot, hold on. Oh no. Okay, that works.
Yeah, that worked.
Yeah, okay, good. Okay, so I'm gonna do this by telling sort of three short stories. The first is really well developed. It's how microRNAs recognize their targets. And this is something we've been working on for probably 10 years. And so it's really well developed. And most of the ideas are published. In fact, I think all of them are. But it's important to tell this story to set the stage for the next few stories, which are unpublished, but nearing sort of a mature state. The second one being how piRNAs recognize their targets, which are very special targets. These are transposable elements. And they're special because these are targets that are actually probably under selective pressure to escape recognition, which is different than how microRNAs work. And the last I'll touch on is how hepatitis C virus hijacks a very specific microRNA, miR-122, to do its bidding. Right. Okay, so I'll start with this, introducing this paper from Dave Bartel's lab. This is one of my, in a way, one of my favorite papers. What they did was they asked the question of how they basically surveyed small RNAs in a variety of different eukaryotic organisms and found that essentially, basically all animals, as far as they could tell, contain microRNAs and piRNAs. And so the last common ancestor of all animals probably had these two different flavors of small RNAs. The sequences, of course, of the RNAs have diverged a lot over evolutionary time, but the chemical structure of the small RNAs have remained the same. And also the core protein factors that bind and execute the functions are also highly conserved. And so the same core machinery in the very core working in humans is also working in this simple animal, the sea sponge. And so to me, this is really, it just highlights how deeply rooted these small RNAs are in animal biology. Right, okay, but this may be too basic for this cloud, but it's fine. So what these things really are is just, little pieces of RNA ranging from 20 to 30 nucleotides in length. piRNAs are typically a little bit longer. In a typical animal, we'll have somewhere on the order of hundreds of unique microRNA species. So there's about as many microRNAs or different flavors of microRNAs as there are transcription factors about. The best understood function of microRNAs is repressing cellular messenger RNAs. And then if you look at conserved microRNA binding sites, it looks like microRNAs regulate somewhere between a third to half of all mRNAs. And for sure, this has been borne out that knocking out specific microRNAs leads to all sorts of defects in a lot of different biological processes. Again, speaking to the importance and the widespread use of these small RNAs in animals. And then piRNAs are actually even more complicated and I think more mysterious. A typical animal will have hundreds of thousands of unique piRNA sequences. So it's really, really complicated. It's clear that in some cases, piRNAs can regulate gene expression, but the most deeply conserved and most fundamental seems to be silencing transposable elements that are pervasive in animal genomes. And this is really important, particularly in the germline. So piRNAs a major function that's conserved throughout the animal kingdom is maintaining the stability of the germline genome. And of course, this is totally necessary for fertility. Right, okay. So in and of themselves, these things are just little pieces of RNA, when they would have no function at all. And so really what endows them with these regulatory powers is the proteins that bind to them, right? And so for microRNAs are bound by a class of proteins named argonaute, and then piRNAs are bound by a class of proteins, a related class called Pb proteins, right? And so starting with how argonaute works, argonaute uses the microRNA as a guide, and it recognizes complementary sequences and mRNAs through just Watson-Crick base pairing, and then that then argonaute then recruits additional factors and promotes mRNA decay. piRNAs, Pb proteins similarly use piRNAs as guides, also through Watson-Crick pairing. And then once they find targets, they can act in a few different ways. One is to recruit enzymes or other factors to the genetic loci producing targets to promote histone or DNA methylation. And then the other way piRNAs go is they can directly cleave the target RNA that they've bound to. So these are the modes of silencing, the basic general modes of silencing for these two types of RNAs, right? And so a question that's come out in the field, and certainly in my work, has been really, what is the mechanism that determines target recognition? How do these proteins use their guides explicitly? And then a second question, it's more specific for this talk, is there a difference between microRNAs and piRNAs? Or are they the same? Or do they have functional differences that would reflect their different roles in animal biology? And so I'll talk about that as well. So we'll start first with looking at the mechanism that determines recognition. We'll start with microRNAs. I mean, in one sense, it's sort of obvious. It's going to be base pairing, and that's the main driver. But it's not exactly that simple, right? So a lot of work from the Zamor lab and Bartel lab and many others have shown that microRNAs can be divided into sort of little domains within them. Not all nucleotides are the same in terms of target recognition. In fact, one part of the microRNA is most important, and that is the seed region. So counting from the 5' end nucleotides 2 through 8. This is the primary determinant for binding a target RNA, and it's generally sufficient for recognizing a target. If you scroll down a little bit further down the microRNA, there's also the 3' supplementary region, which is nucleotides 13 through 16. And these are been shown to be able to add to targeting that was initiated by the seed region. And of course, these very strange patterns arise because it's being mediated by the argonaute protein. And so now we move to a structural question, is what is argonaute doing with this small RNA to create these different regions in these domains and then enable the target recognition? Okay, so this is where we get into the structural realm. This is where I feel most comfortable. So this is a crystal structure of human argonaute 2. The protein shown in gray and the microRNA is shown in red. This is sort of a composite structure. We have a lot of structures now. And so we can sort of piece them all together to get a full picture of what the microRNA sort of looks like. Often different parts of it are disordered and different depending on the sequence. But this is generally how it works. And so the 5' end is bound on the left, and the 3' end is also bound on the right. Right. And so, I mean, argonaute proteins and Pb proteins, all these different domains with these different names. And these are important to know. There's also, even beyond the names of these domains that were identified long ago, there's also structural features that become clear. And the one that's going to be important in this talk is this thing called the central gate, which sits right in the middle of argonaute. But as a structural biologist, I actually don't do very well with names. And so we're going to try and strip all that away and just think about the molecules just in terms of their shape. Right. And so instead of showing argonaute as a ribbon, I'll show it as the surface of the molecule. So this is the surface of argonaute. I made it sort of translucent so you can see the RNA, sort of the microRNA running through it. And so now we're in a great position. So we can turn it and we can look at it. So we're looking down, you can see there's the microRNA running through. We can even slice into it. And you can see argonaute basically has different regions inside of it and creates two chambers, really. One is the seed chamber, where the seed region of the microRNA resides. And then over on the other side is the supplementary chamber, which is about where the supplementary nucleotides reside. And in the middle is this central gate. And the central gate is a funny place because it's the part of the protein that is the most narrow. I can show you this by turning it sideways here. I can't tell. Right, yes, it's very narrow there. It's so narrow that when the protein's closed, which how it seems to spend most of its time when it's not engaging a target, it can just barely thread the single-stranded microRNA through the middle. Right. Now, adjacent to the central cleft. On the right side is the supplementary chamber. Here we're showing it from a side view. And what's interesting about this region is that it's also kind of narrow. And that results in, if you look at the supplementary region of the microRNA and sort of just strip everything away and just show these nucleotides, you can see there they are, 13 through 16. You can see they're all split out in different directions. And so basically in this conformation, it's going to be impossible for the supplementary region to engage a target RNA through base pair interactions because it's not physically possible. And of course, the reason it's sort of squished like this is because it's in this conformation, the supplementary region has to fit through this narrow little region here. And so it has to be sort of splayed out, right? And so we leave from there and we slide on over to the seed chamber over here on the other side of the central cleft. And that's shown here, and you see it's much wider. And this, you can see the seed region here. I can strip away the protein. You can see that's the microRNA seed region, 2 through 7. So what's important here, and really important, this is unknown for a long time, is that the way Argonaut works is it pre-organizes the seed region in a helical conformation. And this has been shown to then increase the affinity for target RNA substantially by paying the entropic cost of forming a duplex. It's a lot like how locked nucleic acids work. Right, so that's very important. All right, but if you put the protein skin back on, what you can see is even though the seed region is pre-organized, only a few nucleotides are actually available for pairing initially. So really, a lot of work from this is from Phil Zamore's lab and Trilman Ju's lab show that for the most part, initial recognition of target RNAs starts at the 5' end of the seed with these nucleotides that are exposed. And I think that's pretty well worked out. And so we can start talking about target recognition. It could be a lot of Argonaut crystal structures. We'd probably determined, I don't even know, 24 by now. And so we can, so there's our initial target, initial sort of part of the seed region poking out for target recognition. We can dock. We think target recognition looks something like this. This is based on real crystal structures. But if you see in this initial conformation, it's impossible to have any base pairing beyond this first part of the seed. But I can interpolate then to a structure where the target's fully bound. And what you'll see is Argonaut then opens up, and that then provides space to have full seed pairing. Right, and this is great. So we can peel away the skin. You can see that is the seed region, and it forms this nice duplex, 7 base pairs long. And then immediately downstream of the seed region is the central region. And what you can see is it's impossible for the pairing to continue beyond the seed into the central region. And that's because nucleotide 9 of the central region is basically slid over and sits underneath the very end of the target duplex, right? And so we can't extend the duplex any further. And of course, the reason for this, the reason the central region is all squished up is because it's sitting in the central gate, right, in that narrow point. And so it's just too narrow to have pairing go beyond that point in this conformation, which seems to be a really well-populated low-energy conformation for the protein. And so what this means is that the seed region, which was defined a long time ago by Ben Lewis, Dave Bartel, and Chris Burge, and many others, but really a lot of people worked on that. But the reason it's 2 through 8 is physically defined by the size constraints of the seed chamber. So Argonaut creates a chamber that has a specific size, and that makes the seed this exact length. And that's why that happens. Okay. So moving on, if we shift over to the other side of the central gate, we have the supplementary region. Now in this conformation, once target's bound, you can see the supplementary nucleotides are all splayed out together, and they're actually ready for target binding. And the reason for this is because the 2 chambers are connected. And I can zoom out and show that here. So here we have on the left, the seed chamber, on the right, the supplementary chamber. And the thing in between them is the central gate. And so the idea is if you have seed pairing, that drives open the central gate. And that then opens up also the supplementary chamber as well. I can show that by if I remove the target RNA and then let the protein collapse back down to the conformation before target binding. You see the seed both chambers collapse, and then the supplementary region gets sort of squished down to that narrow region again. If I go back up, you can see it opens up, and now you have space for target pairing. So a target comes in and it binds just like this. And that's this is the real crystal structure. And that's basically how we think it works. Right. And so defining the supplementary region in sort of rigid way, as we did for the seed region, in a sort of structural way, is more difficult. One thing is that, so there's the seed pair and the supplementary. And in this conformation, the 2 RNAs, target and microRNA, do not wrap around each other. Instead, we have this unstructured bridging region, which in vitro can be from 3 to 10 or even more unstructured nucleotides, and you'll still be able to form the seed and supplementary paired regions just fine. Right, and so the distance on the target doesn't have to be fixed between these two regions. So that's one challenge. And the other part is defining exactly where the supplementary region is in the microRNA. And the thing we noticed was that in this conformation, the 3' end also remains anchored, right, to the, this is in the PAS domain. And what that means is there's actually a set number of nucleotides, in this case it was 5, between the 3' end and the supplementary region, right? And so also when we look at the structure, we notice that when for the molecule to open up, because the 3' end remains bound, that actually pulls the entire 3' tail into this extended conformation. I can show that best if we sort of move to a ribbon diagram. So here you can see this is the 3' end bound at the top there. If we move down to the closed conformation, you can see that the 3' tail can relax. But if you move back up again, it really pulls it, almost looks like it pulls it tight. And so this raised the question, is there tension in the 3' tail that happens when the molecule opens? And if so, does this dictate opening and by extension target affinity? Because opening has to, which is accompanied by target affinity. So it's the experiment we did. This is the only experiment I have for this part of the talk. As you tested the idea, we made 3 different complexes of argonaute-bound microRNAs. Everything was identical except the length of the microRNA. So we made one that was 21 nucleotides, 22 and 23, always extending off of the 3' end, which doesn't match the target. Then we gave these complexes target RNAs, and we just measured the rate of dissociation. And what we found was as we increased the length of the 3' tail, presumably creating more sort of slack in that region, then the dwell time of the target on the complex went up. So the protein could bind more stably when it has a longer 3' end. And I don't know if this happens in cells, but it's clear that 3' isomers are prevalently found in nature. So this might actually have a biological function. We also know it seems to be primarily functioning through the supplementary region, because if we do the same experiment just using seed pair targets, everything collapses back down. There's a small effect, so you can get better seed pairing, even seed pairing with a 23 nucleotide microRNA, but it's not the same sort of really big effects we see with the supplementary region. And then the last thing we asked is, what if we shift the supplementary region, right? And so these targets were all pairing, had a couple complementary to 13 through 17. What if we shifted it over to make it 15 through 19? And then we do the same experiment. And this really showed the effect strongly, where the 21 nucleotide microRNA basically treated this target like a seed pair target. It released it with a half-life of 3 minutes, pretty close to seed pairing. But as we increase the length of the tail, that then allows the microRNA to use this sort of shifted part of the tail. Right. All right. And so this comes back to, this is a small detail, but I think it's interesting, how we define the different domains of the microRNA. And so the classic way of thinking about it is you start from the beginning, right? You start from the 5' end, you measure 1 nucleotide, and that gives the seed region. And you measure 4 more, and then you have the supplementary region, and it makes perfect sense. But I don't think that's how Argonaut works when you look at the structure. I mean, I think it's half right. The seed region's right. So for Argonaut, it definitely anchors the 5' end, and it counts, and that's the seed chamber, and that's defined. But for the 3', for the supplementary region, I think it counts backwards, because the 3' end is also anchored. And so in the structures we have so far, it's 5 nucleotides, and we don't know if this is sequence specific, but we'll say for now 5 nucleotides, and then counting back, and then you have the supplementary region that fits in that region there, right? So it's not a huge change, but it is I think it could be interesting, right? Okay, so now we have a pretty good idea for how microRNAs work. As I said, we've been studying for a long time. This is sort of a synopsis of all the papers and the structures that we've done. So we have a lot of understanding. Now the question becomes, what about piRNAs? A lot less is known, right? Okay, but we're not totally unknown. So we do know that the seed region is important, just like microRNAs. And this is because all the targets that have been identified have strong seed pairing. We also have crystal structures of Drosophila peewee and the silkworm seaweed. So we have an idea for what peewee proteins look like. And the answer is they look a lot like Argonaut proteins. But what we don't have is everything after that. how does the protein move? How does it open? What does it look like when it binds to a target RNase? How does it recognize these things? And the reason we don't have this information, actually, we don't even have basic biochemical binding data. And the reason is because we don't have a system, a purified system or a way of making purified Pi protein bound to a defined pi RNA. And so we started this to try and address this issue several years ago. I looked for a recombinant source of Pi proteins, and we tried human first, and that didn't work. The proteins just weren't expressed very well. But then we said, well, every animal on Earth seems to have these things, so why limit ourselves to humans? And so we just started screening different animals until we came across one that we could express and purify and really use in vitro. And this happened to come from a very ancient organism, the freshwater river sponge. And so we're really lucky to come across this construct. All right, so this is just basically very quickly showing how we do the experiment, how we do the screen. Basically, we make the recombinant protein in some form, we get it to a sort of partially purified form, and then we add to that a synthetic pi RNA, and we ask we ask the question, if it's assembled properly, that should be able to recognize a target, and the target will be then immobilized. So we should be able to pull down the protein if it's functioning correctly. And so this is a typical result. This is a protein gel. So on the left lane is the input protein. You can see there's a prominent band for the Pi protein, and then everything after that is pulled down, is what was pulled down under different conditions. You can see only when we added the pi RNA that was actually complementary to the pulled down oligo, we pulled down the protein. So it looks like it was working fine. So that was a nice result. Right, and now you have this thing, you can do all kinds of stuff. So one thing we did was we determine the structure. So in this case, we used cryo-EM, and this is the actual original data. You can show the pi RNAs here shown in red, and I can sort of turn everything gray to show that. So we could see the 5' and 3' ends are really well defined in the electron density, but the middle part isn't. We can see sort of ghosty density in the middle, so the pi RNA is running through the middle, but it's not taking a defined structural conformation, so we can't really see it, right? I can just rotate the molecule here. This is just to show this with the quality of the data. I think the structure was 3.6, 3.8, something like that, angstrom resolution, which luckily, since we have the Pi structure and the Cwe structures, we can build an atomic model that's pretty accurate using the data and those structures. And so now we're in great shape because we can build an atomic model using these data. Right, and now this is great because now we have a structure of Pi, another structure of Pi protein, and we can start doing comparisons. And so this is a side-by-side comparison. On the left is crystal structure of AGO2. In the middle is the crystal structure of Cwe, bound to a mixture of co-purifying pi RNAs. And on the right is the EM structure of Pi, the sponge Pi, bound to a single defined homogeneous pi RNA. And I would say from the sort of 100-foot view, they all basically look the same, right? I mean, all of these domains, and they'll kind of fold together, and the guide RNA runs through the middle. But there are differences once you start to look closely. And so the first difference that was really glaring to us was Argonaut pre-organizes the entire microRNA seed region. I can sort of highlight that here. It turns out this is not the case for the Pi proteins. So all the structures, there's 3 Pi structures now, and they all have partially ordered seed regions. And so I can show that here. So this is, on the left is all the contacts AGO2 makes to pre-organize the seed region of the microRNA before target binding and sort of hold it in that conformation. You can see there are very many. And on the right is what the sponge Pi does. It basically, only the first 3 nucleotides are ordered, and after that is disordered, and there's far fewer contacts, right? And so this raises a sort of obvious idea that maybe pi RNA seed region is not the same as a microRNA seed region, not functionally. And what's great is we have this recombinant system, so we can actually directly test this question. So we made complexes of human AGO2 or sponge Pi loaded with exactly the same guide RNA, and then gave it a target RNA that matched the seed region, and then we could just measure affinity. And the result is that the Pi protein made a seed region that was 150 times lower affinity for the seed match target than Argonaut. And we did it again also with a longer guide RNA with a 3' with a 2' methyl on the 3' end to emulate a pi RNA, and basically the result was the same. So the conclusion is that Pi creates a much weaker seed region. So pi RNA is a weaker seed region than microRNAs. And it looks like this is, look at the conservation of these residues that make these contacts. This might be the case for all Pi proteins in general. Right, so that's the first thing. Then the second thing is we start to move downstream of the seed region, and we look at the central gate, which we've described in detail. In AGO2, it's really narrow, but in PIWI, it's different. So the same residues that make that yellow bit, that yellow loop that sort of makes the top part of the central gate in AGO2, those residues in PIWI proteins are folded into a little helix, and that's shifted over to the left towards the seed region. And so for now, we're calling this the seed gate, because I think it actually is involved in recognizing targets in the seed region, but I'll get to that later. Right now, I want to talk about the central region. So of course, the central region is very narrow in AGO2, but because of this relocation of the same residues and sort of a rearrangement of the domains, the gap is wide in PIWI. And so this raised the idea that maybe downstream of the seed is different between microRNAs and piRNAs. And so again, we can test this idea. So we made a complexes with the defined piRNA. I have to see, I can't see. Okay, sorry. And then we bound, sorry, I'm doing something funny with my screens. We bound, first as a control, we bound a target RNA that just matched the seed. And then we basically measured the release. Right. And so for AGO2 releases this little target RNA that matches the seed over the course of minutes, right, as expected. PIWI released it over the course of seconds, really fast, which makes sense, which fits with this sort of idea that piRNAs have a weak seed. But now what we can do is we can make the RNA longer. And so in this case, we extend it into the central region, going to 2 to 10. We do that, AGO2 treats it exactly like the seed matched RNA because it doesn't use the central region, as we knew. But this is not true for PIWI. PIWI actually binds much tighter to the extended RNA. We can go further to the full central region. Again, AGO2 treats it like a seed paired target, but PIWI binds much, much longer. And it's not until we extend out into the supplementary region that AGO2 can start to see an increase in the affinity for the target, but PIWI is way ahead. And so this really two ideas come out of this. First is that even though PIWI has a weak seed region, it can compensate for that by adding in additional downstream pairing. And moreover, because it can do that, PIWI can bind target RNAs with a much, much longer half-life than AGO2 can. And so I think piRNA is going to recognize that we can bind to their targets and stay bound for a very, very long time. With some of these, we make extended targets, the half-life can be up to 8 hours long, where Argonaut's usually off, in a few minutes. Right, okay, so we see these clear differences in target recognition, but for structural biology, we want to see what it looks like. And so we also looked at structures of the PIWI complex bound to the different target RNAs as well. And so this is one where we had, this is probably the most successful one, where the target RNA spans from the seed to the supplementary regions from 2 to 16, right? And so this is the density shown here. The target RNA is shown in blue, and you can see it's wrapping around the piRNA in red. This is just to show the quality of the map is really nice. It's like 3.4 angstrom resolution. So we could build everything, well, almost everything. And it's very, very satisfying, right? Okay, so it's piRNA, target RNA. So we can build a model and look at target and build a top model. So this is really great because now we can start to look at how PIWI actually engages the targets that it recognizes. And so I'll do this. This was hard to do. I was working on these slides last night. And it's kind of hard to do. If we'll go back to our idea of just looking at the shapes of things. So that's the shape of the PIWI protein and then the target RNA. And the RNAs are bound through the middle, as you can see. I'm going to remove the target RNA. And we'll just start first by looking at the piRNA. And we're going to zoom in and look at the seed region. So what you can see is once the target RNA is bound, PIWI makes extensive contacts along the piRNA backbone through the seed region. It also, if we put the target RNA back on, PIWI also makes contacts to the minor groove that's formed by the duplex, between the piRNA and the target. I can show that here if I sort of show the surface of the duplex. So this is just in the seed region, and you can turn it around here. And so basically the minor groove is the region between the two strands, sort of shown there. And so the important thing is that PIWI probes the entire minor groove. And so the idea when you look at this structural biologist is the combination of all these backbone contacts, and most importantly, the minor groove contacts, means that PIWI should be really sensitive to mismatches, or should be able to sense mismatches in the seed region. Because once you have mismatches, the bases start sliding around past each other, and that distorts the minor groove substantially. Right, okay. Now, if you look downstream of the seed, it's a very different story. The pRNA makes almost no contacts to the protein. Instead, all the contacts are to the target RNA. And this has to do with just the helical nature of the pRNA target duplex. And so so PU makes multiple contacts along the target RNA backbone, but most importantly, it doesn't contact the minor groove. So if I turn the model here, you can see there's the minor groove running there, and there's basically no contacts to the minor groove beyond the seed region. And so what that means is PV sort of breaks the pRNA into two parts. On the left, we have the 5' half, which includes the seed region, which has all of these contacts. And so we anticipate it should be really sensitive to mismatches. And on the right, it has contacts to the backbone, but nothing to the minor groove. And so it should be able to tolerate, much more tolerant of mismatches at the second half. And so the great thing here, again, is that we can test it, right? And so the way we do it, in this case, what was most illuminating, we made the complex of PV bound to a pRNA, gave it a target RNA, and then just measured the rate of target binding. And then we did this for a bunch of different target RNAs, which contained mismatches, 3 nucleotide mismatches at different regions, right? And so spanning the entire target RNA. So the mismatches are represented by the lack of pairing, right? And the answer is, by and large, PV isn't bothered by the 3 nucleotide mismatches. Most of these targets bound at basically the same rate as the target with no mismatches. The only exception was in the seed region. Right, exactly as predicted. So we can say that pyrite targeting is sensitive to mismatches in the seed region. And this, of course, was not a huge surprise because the targets that have been found have seed pairing, have seed complementarity, and the structure really suggested it. So that wasn't a huge surprise, but it felt good. But what was a surprise, I think more interesting, was that targeting was particularly sensitive to mismatches in the 3' half of the seed. So these 3 mismatches dropped target binding rates by over 2,000 fold. So it's almost, I'm not even sure we could measure the target binding rate because it's so slow and so weak. So we estimate it's 2,000 fold. And this was really interesting, and it's different from Argonaut. We know from work from Phil's lab and Schroman that the most important for an Argonaut for initiating for target recognition is the 5' half of the seed. And so we wonder what's so special about PV. It might be how we're doing an experiment, but I think there's a little more to it. And that has to do when we look back at the structure. So this is the structure of PV bound to the pRNA before target bind. I can zoom in on this near the seed region here. And then, okay, that's the nucleotides 2, 3, and 4. And they're poised for recognizing their target. And then the rest of the seed region, of course, is unstructured prior to target binding. And so I can then put on the target RNA. You can see that's the same problem as Argonaut. It can't go any further without a conformational change in the protein. But in this case, though, the conformational change needs to happen in the seed gate, which is, Argonaut has a similar structure, but in PV it's expanded because of those residues that were in the central gate have been shifted over. And so our idea, so I can interpolate from this structure to the actual target bound structure, and we'll see as the seed gate will go up, and that actually opens access to the entire central cleft. So I'll do the interpolation now. You can see it going up, and the entire central cleft then opens up, right? And that then provides space for base pairing at the 3' half of the seed. And the idea here is what the seed gate does is it actually probes the minor groove of the 3' half of the seed. So our model is that if you have proper pairing in the 3' half of the seed, that basically enables the pRNA target duplex to hold the seed gate open in this open conformation, and that then allows base pairing to propagate into the central cleft. And without that, you can't get past the seed gate. You need that pairing. That's how we explain it. It's a nice idea. It's hard to test, but the best thing we could come up with was making a mutant. So on the left is the wild type PW, and on the right is a mutant PW where we removed the seed gate. We just added a little flexible linker in between to tie things together. And then we went back and measured target binding. So for reference, this is how the wild type binds. It really suffers. I'm showing mismatches at 2 or 4 or 5 to 7. It really suffers when you have mismatches at 5 to 7. But if you look at the seed gate mutant, you remove the seed gate, that restores target. It basically doesn't change target binding very much, except for the target with mismatches in the 3' half of the seed. The binding rate goes up a factor of 14. So consistent with the idea of once you remove that barrier, those mismatches aren't so costly anymore. Right, okay. And then the last thing we looked at for PW was this target cleavage activity. Now this we struggled with a bit because our PV preparations that we make, they have this target cleavage activity, but it's not particularly, it's not wild, it's pretty weak compared to Argonaut. But we could see some, and so we could characterize it. And so I should say target cleavage is a really important part of PV function, because oftentimes what happens is when PV cleaves a target RNA, the cleaved target itself can then be transformed into a new piRNA, and the entire, and through an amplification process termed the ping pong cycle. And so this is how piRNAs can, a really important part of how piRNAs can adapt to the sequences they're trying to silence. Right, and so we wanted to understand cleavage activity. We did a lot of different experiments. Probably the one that was the most illuminating was when we looked at the amount of complementarity that was required to see the cleavage activity. And so this is a denaturing gel showing target RNAs are all the same length, but they have different degrees of complementarity to the 3' end of the piRNA. And what you can see is if you pair just a 2 to 15 or 2 to 16, the cleavage is really, really weak. And it's not until you go out to 18 that you start to see cleavage activity. And so what's interesting about that is that we know from our structures that getting out to 18 means you're beyond the central cleft. So you really, it seems really to extended duplex to have, to start to see this cleavage activity. In contrast, just for comparison, AGO2 cleaves all these RNAs really, really fast. So cleavage under our conditions by PV is a lot slower than AGO2. And so, but we do know we can stimulate it by extended pairing. I don't exactly know why we need extended pairing, but we have some ideas. And that came from our efforts to look at structures of the complex with extended complementarity. And these initially were sort of fraught with trouble and failure. And so on the right is the 2D class averages that we'd see. And it was very puzzling when we first saw these. This is when the piRNA, I think complementarity extends all the way out to 25 of the piRNA. And you can see that density right there corresponds to the piRNA target duplex. That's definitely an RNA duplex. But the weird thing is it's extending away from the globular protein. This seemed impossible when you're looking at 2D, because we thought the RNA should be encased within the protein. But once we determine the structure, it's a difficult structure, so we can only get 8 angstrom resolution. We could get a reconstruction at 8 angstrom. It looks something like this. And when we try and fit what this is, it looks like it's half of the PV protein bound to the piRNA target duplex. And the half is the mid and PV domains. And this is the part that binds the 5' end. And so the idea for what happens is once you extend past the central cleft, that pushes PV into a really open conformation such that the other half of the protein becomes disordered, and you can't see it anymore in these class averages, because these averages are averages of hundreds of different molecules, images from hundreds of molecules. And so anything that's moving around gets averaged away. But we can dock onto this structure, the structure we had before, where pairing is limited to the central cleft. And you can see that's the part that's disordered. And I think this is right, because if you look at interactions between the disordered part of the protein and we know where the target RNA has to go, there's all these steric clashes. And so it looks like somehow this extended pairing forces the molecule into extended conformation, and that at least coincides with the activation of the cleavage activity. Right, okay, so the question, are microRNAs and piRNAs the same or distinct? I would argue they have similarities, but they're actually quite distinct. And I think that these distinctions do in fact arise from their different roles. I think these actually would allow them to have different roles in animal biology, and I'll explain. Okay, so Argonaut uses microRNA, and it creates a very strong and precisely defined seed region, right? It's just 2 to 8, and that's it. And so what this means is a single microRNA can repress hundreds of different mRNA targets. And that works really well, because you have this really small and defined recognition element. There's also, you can also tune it a little bit. You can tune affinity by increasing affinity a bit, by increasing supplementary pairing, and also perhaps, at least in vitro, by changing the microRNA length, the 3' end, we can increase affinity further still. And so it's possible there's a tuning of microRNAs, and that raises the possibility that there might be special targets that for whatever reason have higher affinity interactions with the complex. But that is something that would remain to be determined. But the most important thing is this strategy allows microRNA to repress many, many targets. Of course, this would be a disaster for piRNAs, because if piRNAs had a strong seed, given the diversity of the RNA sequences, they would target everything, right? And it just wouldn't work in terms of specificity. But of course, the cell wants to be able to have a large RNA repertoire so it can be prepared and tackle all these different transposons counters. And so the way that evolution or PB works is it still uses a seed region like Argonaut, but it creates a weak seed so that pairing it's not as what's the word, promiscuous, right? But you can still have strong selectivity by making the seed gate require seed pairing, right? And so this gives you selectivity in the absence of high affinity. It's very clever. And then beyond that, you have an open central cleft that allows all different types of base pairing. As long as you have a more or less helical structure, PB will bind to it, it seems. And so you can, and what that means is beyond the seed, PB's tolerant, this open central cleft is tolerant of mismatches. And basically, the idea that we have is that this might allow PB to then be flexible in recognizing related or evolving transposons or transposons that are under, pressure, selective pressure to escape recognition. PB can sort of follow them. But then for cleavage, what we see is the requirements are much more stringent. We need much more pairing, and it has to be longer. And this might reflect the fact that once you cleave a target RNA, you commit now to making new piRNAs. Right, so that's the difference between piRNAs and microRNAs. Okay, and the last part of my last little story, I've totally lost track of how much time I have, but hopefully we're okay. Okay, is how hepatitis C HCV hijacks miR-122. This is also unpublished, and there's a lot that we're trying to do in this space. It's very complicated, but I'm really interested in the general question of single-stranded RNA viruses. So these many single-stranded RNA viruses, obviously. And what's interesting about them is their genomes are actually targets for small RNAs, just naturally by their single-stranded nature, right? And so I think this is a really underdeveloped and underexplored area of small RNA biology that I think is really interesting, but it's not totally undeveloped. Probably the most best understood seminal work from Peter Sarno's lab was in hepatitis C virus. So hepatitis C contains the genome of the RNA genome contains two binding sites for a microRNA 122. So miR-122 is a liver-specific microRNA, which is of course where the hepatitis C infects. And so what's interesting here is there's actually a lot interesting here. There's the two binding sites for miR-122 are right next to each other on the very 5' end of the viral genome. And they're upstream of the iris, which is involved in initiating translation. So these have been known about for, I don't know, about 15 years, and they've been studied pretty extensively, and they're still sort of mysterious, but it is clear that binding of Argonaut and miR-122 to the 5' end is important for maintaining viral RNA stability inside infected cells. It also seems to be important for folding of the 5' UTR, and it also seems to in some way regulate translation of viral proteins. And this is actually a major problem, major challenge that viruses have because their genome is a template for both transcription or translation and replication, and these two things are opposing forces. And so there has to be a balance. And so it's pretty clear that miR-122 plays a role in regulating translation and that balance. And so we have ongoing efforts to understand all of these things, and they're in different phases of development. The part we understand the best is viral stability. That's the easiest thing to test. And so that's what the part I'll talk about, sort of closing up this talk. And so what we found is the most important part for viral stability is the binding of the first Argonaut, the very, very 5' end of the viral genome, the genomic RNA of the virus. And what was really interesting about this is when we looked at the structure, so this is just a schematic of the viral RNA structure, there's a stem loop structure, SL1, stem loop 1, right in the middle of the microRNA binding site. And so this totally contrary to what we normally think about microRNA target sites, they should be devoid of structure. But here we have a very strong GC-rich stem loop. And so we are eager to see what that looks like. And this is what we'll end on. So this is the structure of AGO2 ternary complex bound to miR-122, and then the 5' end of the genomic HCV RNA. and it's pretty wild. So there's the 5' end and the 3' end of the microRNA both anchored as before, and basically exactly the same conformation. The 5' end of the viral genome is here over on the right, near the supplementary region. And I can sort of strip away the protein, and you can see what's happening here is the two RNAs are forming a three-way junction, right? And so the seed region is shown here, paired to the viral RNA. On the other side, the viral RNA pairs to the supplementary region. And in the middle is that stem loop 1, the stem loop structure. It's so interesting. So you didn't know this, but Argonaut can actually, what this shows is Argonaut can accommodate a three-way junction inside of it. And I can show you how that works by putting the surface of the protein back on. So we can look down on the surface of the molecule from the top view, and you can see that stem loop 1 basically is wedged right in the middle of the central gate. And this pushes Argonaut into this open, extended conformation that we rarely see. And if you look from the side view, you can see it fits perfectly. There's so many interesting things about this. What you can see is the top part of the stem loop is open to the bulk solvent. So there's no limit on how far it could extend. So these three-way junctions could be much bigger parts of RNPs than just this small limited RNA, just in general for Argonaut binding. I can show that if I show that's just a ribbon diagram showing here. And there's the way that the viral RNA fits inside Argonaut, it's almost like a lock fitting inside of a key. Down to these little details, like there's this little binding site, a very specifically placed target nucleotide binds in this target nucleotide binding pocket inside Argonaut that we had no idea existed. And so when we look at the structure, what it looks like is this viral RNA is popping Argonaut into this really extended conformation and basically trapping the protein complex on the very 5' end of the viral genome. And so I can show you that if you flip it around here, this is the 5' end of the viral genome, and you can see it's tucked away safely inside this sort of expanded supplementary chamber. And so the idea is when it's bound in this way, it's protected from exonucleases. And so we could test that idea by just using it, again, in vitro biochemistry as we do. And so the main exonuclease that's thought to degrade the viral RNA and is exon 1. Right, and so we can set up an in vitro assay where we transcribe the entire 5' UTR of the HCV and then add in miR-122 with AGO, let that bind, and then treat it with a purified exon 1. And we do that when Argonaut's bound, we can put in a lot of exon 1, and the viral RNA is pretty stable over time. So it's a fraction of the RNA degraded over an hour. You can see it goes down, but it's pretty stable. For comparison, if we make a mutant where we eliminate the first binding site, you can see it's destroyed in a few seconds, right? And so now these are the two extremes. And somebody asked the question, do you need that stem loop? Does that matter? And the answer is yeah, it seems like it does. If you remove stem loop 1, the stability of the RNA in the presence of exon 1 goes down substantially. And we have results transfecting into mammalian cells showing this is probably true in cells as well. Right, and so there's a lot more going on here, but I'm gonna stop there. All right, so this comes to the end. So the summary, really, so the structural basis for microRNA targeting we worked on for a long time, and the most important thing is that Argonaut creates a strong seed that allows it then, and a really defined seed that allows it to go after many targets. The effects of the 3' isomers, I'm very interested to know if this plays out, if these affinity differences we see by extending the microRNA tail, if that also plays out in a biological context. For pRNA targeting, the important thing here, I think, is that PIWI creates a weak seed, and that allows higher selectivity, but then it balances that against this tolerance for mismatches, and so it can make it flexible for recognizing transposons. And then this work that's just emerging from the lab is the idea that RNA viruses, in this case HCV, can use RNA structure to exploit AGO2 and push it into conformations that we don't normally see. In this case, trap it. But we've looked at other RNA viruses, and so Zika has a similar motif, but it's internal, and I don't know why, but it's interesting. Yeah, and I'm actually curious to know if these type of sites that we see in HCV also exist in some cases in cellular RNAs, but that remains to be determined. Okay, and so I end with acknowledgements. The AGO2 team is basically shown on the left. In the middle is the PIWI team. On the right is HCV. Down below that is all the sort of friends and colleagues that gave us all the different PIWI constructs that we asked for over different times. And below that is all the funding sources. And that's it. That's my talk.
Fantastic. Thank you very much, Ian. It was great. As I've just written into the chat, feel free to type your questions in there or unmute yourselves and go ahead and ask your questions. Maybe we'll start with everybody unmuted.
Ian, that was a great talk.
Well,
I had two questions for you. Does their river sponge peewee have symmetrically dimethylated arginines in the N-terminus that matter in this structure? And then the second question is, have you tested artificial piRNAs lacking 2'-O-methyl, and do they affect some of the kinetics you see?
Yes. So sorry, those are good questions. Yes, it had a big long arginine-rich tail on the N-terminus, and the construct we ended up using was truncated most of the way through. That was a decision based simply on what gave us the best expression. But we've done some structures with other argonauts with the tail there, and you can never see it because it's unstructured. So, but yes, in the sea sponge and the river sponge, yes, they have this just like in mammals. And we have tested the 3' modifications, and we see a moderate effect. It's not very strong. The effect, a couple fold. It depends on the target you're looking at, but sometimes you can see an effect. But in general, we gave it like miR-122 or a big piRNA, and it behaved more or less the same.
All right, cool. Thank you.
Hi, I want to ask a quick question. This is a fascinating talk. Thank you so much. When the stem loop holds the central chamber, the central gate open, does it do the same thing like in like a peewee situation where it makes the supplement, the 3' end, have a more sustained sort of promiscuous binding and longer binding?
Yeah. Oh, yes. I didn't talk about this. This is right. So this was really satisfying. It turns out it had been shown by Stanley Lemon, who's an HCV guy, that hepatitis C infection actually needs the long isomer of miR-122. It needs the 23-mer. And then we found that you can't get to that extended conformation unless you use the long isomer. It was actually his paper that helped us get the crystal structure, because we couldn't get crystals because we were using the using a 22-mer. And so just by increasing the length one nucleotide longer, that allows it to open. So yeah, you need the longer isomer for that.
Ian, great talk. Where to start? Let me. So this three-way structure that you show with the HCV, that you show that it protects more when you have the first stem loop rather than when you don't have the stem loop. Could it be that you have accessory proteins binding to the stem in conjunction to AGO in that protrusion that you show?
Yeah, I think it's possible, and it's something we've thought about. I think there is space to bind onto the stem loop, but I don't know what protein that would be. So, I mean, we could make something fake, like we could put a U1A stem loop there and we could bind something there just to show that it's possible. It's really interesting motif. So if you set it up just right, you have to have the position of the stem loop exactly right, and you have to have that base that was sort of sticking to the protein. That has to be a purine. But if you do that, you can have even just two RNAs that aren't even connected to each other. If they're anneal and form that junction, argonaut will bind really tightly to it, which makes me think it's probably something that happens, even outside of the context of the virus. I suspect the virus discovered this and is now showing it to us, as viruses often do.
And if I may, another question, that extended target dissociation time that you show when you have a 23 mature microRNA, is it translated also in enhanced repression of the target because you have extended binding?
Let me do this. We haven't done that experiment, or maybe we did and it didn't work. I mean, we're pretty lame at doing biology. We use like luciferase and stuff. So I wouldn't trust our results. I would expect that it would. I mean, everything. So Dave Bartel and Dave's paper was really great in showing that the affinity does matter of the microRNA does correlate with the degree of repression. And so I would expect by that reasoning, it would make a difference. As long as there's not some threshold you pass where more affinity doesn't do anything.
Thanks.
We actually have a question from Liang Hung, who asks, what are your thoughts on AI such as AlphaFold that can help predict structures and interactions of proteins and RNAs?
Oh, that's exciting. Oh, I think it's great.
Do you use it?
I think, I mean, I've never used it, but I'm excited about this. I think it's a very exciting development. I think it's been a long time coming. And I don't know, does that mean I'm out of business? People talk about that. I don't think so. I just think it'll shift the questions we ask. If we can just predict structures straight up, I think that'd be great. I still think it's a little bit of ways from, I think it can find, domains and even multi-domain proteins pretty well, but predicting the RNA protein interactions might be a ways away. Part of that's because we just don't have as many structures as we do protein structures to train the AI on. But this is outside my field. My brother actually works in AI stuff, so.
All right, we'll bring him into the field.
Yeah,
And,
yeah, I know. This kid, he's spending his time trying to train his computer to recognize images of Spider-Man.
There's a question from Vignesh Hariharan. He says, what kind of evolutionary pressure do you think would lead to such a stringent RNAi system? In what context would high stringency be more beneficial than conventional AGO2-mediated RNA degradation? I think you addressed that a little bit, but maybe you want to expand.
I think so. I'm just thinking how to, we're talking about well, I should say this. The one thing we haven't talked about is siRNAs, right? That's the other type of RNA. And siRNAs are sort of harder to define because, if you put an siRNA into a human cell, it'll go into AGO2 and now it's an siRNA, but it's working in a microRNA context. I would say siRNAs are best understood and sort of the cleanest is in flies where there's a dedicated Argonaut AGO2 to mediating siRNA function. We look at siRNAs, there's also a yeast Argonaut structure that does siRNAs. It looks like it's it's something in between, it looks like. It has a really strong seed. It looks like it does at least. And yeah, I don't know, actually. I think we'd have to really, last thing we could do is look at these Argonaut proteins, is look at how siRNAs work. I don't know if that answers the question, but that's where my thinking is.
All right, good. A question from Sean MacRae. How does the conformation of AGO in the miR-122 HCV structure compare to your published TDMD structures?
Oh,
more open.
That's a good question. I think it's a little bit, not quite as open. So they're very close. They're very, very close. I mean, they're close enough that things like crystal lattice contacts start to matter. And so I would say they're nearly identical. And that would then raise the question is, how does it escape TDMD or what's happening there? I don't know. Or why does it elicit TDMD? I wouldn't think so, right? But yeah, so they're very, very similar.
All right, a question from PDZ. Was it T9 that fit into the pocket in the HCV structure?
Yes, it's T9. Okay. That is T9.
And then Peter Wang asks, in your discussion of the propagation of seed pairing in the PV structure, you seem to suggest that the pairing propagates from the subseed towards the 3' end. How does it propagate to such a long helix without the topological constraint imposed by an active 3' nucleotide as shown in your 16 base pair structure?
Right, that's a good question. I didn't even talk about this. Yeah, because in Argonaut, in the microRNA, the two weren't wrapped around each other. But in the Peewee, they're definitely wrapped around each other, no question about it. And so this must mean, I mean, we can't see it, but I'm pretty sure there was a transient release of the 3' end from the PAS domain that had to happen for the two RNAs to wrap around each other. But then in that 16 structure, the RNA is actually long enough that it can come back and rebind after wrapping to the 3' end. And that's what we actually saw in the structure.
All right, cool. There are no more questions in the chat. Any questions from the audience? Just unmute yourself. Actually, we do have one more question in the chat. So from Yanis Flakos, from molecular dynamic simulations we did at some point, we saw that AGO2 loading and microRNA AGO2 conformation could be affected by microRNA sequence as well, but we always use the annotated sequences. Could the sequence of the 3' non-templated additions affect also the effects you see from longer sequences?
The 3', yeah.
I guess he's asking about isomers. I mean, people are always using the miR base sequence of a microRNA, but there are many isomers.
Right. I mean, my understanding is the isomers arise post-loading, actually, generally. I think that that's right. And so it might not be a huge issue. The loading is kind of a mysterious process. It's not very well understood at all. I don't know. I imagine the 5' end binds first, but I don't actually know that. And so I couldn't say. I really don't know.
Interesting. All right. Any other questions from the audience? All right. Well, if not, let's thank Dr. MacRae again. It was a wonderful seminar. Appreciate you coming by, and thanks to the audience for sticking around. We had about 120 people on the call, and we still have 83 on the call. So happy RNA hunting to everybody, and we'll see you at the next HIRM seminar. Thanks a lot. Yeah, no, it's great. Bye-bye, guys.
Look at him go.